Okay, can everyone hear me okay?
Yeah, we can hear you well.
Great. Welcome to the Silverstein Foundation for Parkinson's with GBA conference call. I'm Jonathan Silverstein, and this is our first Industry Insights meeting, presentation by senior management, biotech companies working on GBA-PD, and the presentation is to our fellows of the 2023 class and the 2025 class, as well as some invited guests. Since about half of you have not heard about our foundation before, interacted with us much, I thought I'd just give you a quick update. So after a 20-year career in venture capital, I was diagnosed with GBA-PD and decided to start a foundation. We started out giving grants to private biotech companies and academic labs. We made 39 of these over 6 years, and 4 companies actually currently have drugs in the clinic. Eventually, we realized that the drug development was too expensive for the size of our foundation. So we pivoted to support the next generation of great researchers in GBA-PD, hence our fellows program. A 2-year fellowship for postdocs focused solely on GBA-PD. We brought in 11 fellows in 2023. We will announce our 15 additional fellows later this summer. I invited both groups to join us, and some friends of the foundation did join as well. There'll be 9 speakers today, each speaking for 12 to 15 minutes. And if a speaker goes for less than 15 minutes, we might have time for a question or two. Please use the chat box, and we will do our best to get your question addressed. Our goal is through an open science platform to help the industry unlock some of the key questions surrounding the disease. We will publish or present all of our findings in medical journals or relevant conferences. I am proud of this 2023 class. They really worked collaboratively in diverse fields and really brought a lot of value, I think, to the industry. With that, the first speaker is going to be Young Chen from Lysoway. Can you share your screen?
Yes, just give me a second. Can you see it?
Yeah. Yes.
Right. You see it in full screen, right?
Yes.
All right, wonderful. Well, thank you, John, for inviting me, and really happy to be part of this seminar. And I'm Yang Chen Chu, and currently the founder and CEO of Lysoway Therapeutics. So the topic for my talk is development of TRPM1 agonists to treat Parkinson's disease. So John asked me, because I'm the first one, so I'm going to just give a very brief background for Parkinson's disease. There's a lot to talk about, but it's important to point out PD is the second most common neurodegenerative diseases, with about 10 million worldwide, more than 500,000 diagnosed in US alone. If it's counting misdiagnosed or undiagnosed patients, probably exceeding more than 1 million in the US already now. So in terms of risk factors, age is the biggest risk factor. It's like 65-year-old, you got 1% of the population have the PD. And by the time age 85, reaching 3%. And there's a lot of genetic risk factors too. In particular, GBA, Parkinson, and LRRK2, they are most penetrant, genetic risk factors. And one of the main hallmarks for PD is the presence of the intracellular Lewy bodies, which is mainly composed of misfolded and aggregated alpha-synuclein. And the presence of the synuclein is accompanied by multiple cellular pathologies that eventually leads to the loss of dopaminergic neurons in the substantial Niagara region, which is part of the midbrain, as you can see on the right side, these brown colors over here. So PD is not only shortening lifespans, but also reduce quality of life. And speaking of the risk factor, the age is the biggest risk factor. So for Lysoway, when we look at it, we don't look at each individual genes as the only issue. We're really looking at the overall functional decline. What's really most important here? What we believe is that the age-related decline or malfunction of the autophagy lysosomal pathway it's a systematic decline. It's not an individual gene issue because even if all these risk genes we're talking about, they're not really a monogenetic disease per se, right? Because folks don't develop those diseases when they were young. And autophagy, lysosomal pathway is directly contributing to the buildup of the toxic aggregates like alpha-synuclein in the human brain, because it is almost considered as the only way to get rid of these insoluble protein aggregates. When folks get older, these pathways, functions are no longer optimal. And if you look at the genetic risk factors in AD, PD, and FTD, and looking at all these risk genes, they all either directly part of the ALP pathway or indirectly contributing to this pathway. As you can see, GBA is right in the middle over here, part of the lysosome, and you got PINK1 and you got LRRK2. They are all part of the mitophagy or autophagy pathway. So we're thinking when we started this company, we're thinking if we can find a way to systematically restore the function rather than focusing on each individual genes, that would be really wonderful. And the question is, where is the intervention point? And we believe the lysosomal ion channels, that is, sitting on a lysosomal membrane, they are the ideal targets because in normal physiological conditions, lysosomal ion channels act as a decoders of various, cellular signals, including, for example, oxidative stress, starvation, or things in that nature. So they're all kind of, mediated by the activation of different lysosomal membrane ion channels. And in particular, we chose TRPM1 and TMEM175 as our first two targets. TMEM1, it's a calcium channel, which we're going to discuss a bit more detail later. TMEM175, it's a proton channel that is very important for maintaining the pH inside the lysosome. So what we're currently doing now is we're dedicating our effort to develop therapeutically viable TRPM1 and TMEM175 agonist, hopefully to systematically restore the optimal ALP function in the aging neurons and surrounding cells. So at this moment, and after 4 years of formation, and we now have a developmental candidate that are highly brain penetrable for TRPM1 agonist, and then this agonist now we're finishing the R&D enabling studies, and we hope to actually test it in human healthy volunteers early next year. So why TRPM1? TRPM1 itself, it's a calcium channel. It is a master regulator of the autophagy and lysosomal pathways. And if there's a mutual mutations in TRPM1, most of the mutations are frame shift or deletions, so they abolish the channel function completely, and it leads to devastating, mucolipolysis type 4, and which is a very rare lysosomal storage disorders, and it happens, in the young babies when they were really young. It's a very aggressive disease. However, very recently, a family of 4 Iranian siblings, with age of 29, 27, 35, or even 42, they have a much milder mutation, a point mutation, which is very rare. Now they actually exhibit, cognitive decline and abnormal movements manifested by very classical Parkinson. You can almost say they have the Parkinson disease. It's just younger. So it just shows that the linkage between TRPM1 and Parkinsonism it's really quite there. I mean, in the past, people say TRPM1 is not a risk gene for Parkinson disease, but it's not really true. And so if you activate TRPM1, it really initiates multiple downstream pathways. For the longer term, it activates TFAT, TFE3, or MIF, which is a master regulator of a clear gene network, eventually leads to lysosome and autophagosome biogenesis. So, enhance the overall function of the ALP pathway. For the shorter term, it also promotes the autophagic trafficking by including induction of autophagy and promotes the lysosome and or focus on fusion, which is really important for the ALP pathway to go into completion. And thirdly, activation of TRPM1 also leads to lysosomal exocytosis too, which is very important again for waste removal and membrane repair. And overall, altogether, they enhance the waste clearance, resilience to the cellular stresses, and augment function and the cellular, cell survival. So pharmaceutical activation of TRPM1 holds the potential to address, multiple neurodegenerative diseases, of course, including PD here as well. So people noticed that once the TRPM1 was discovered as a calcium channel in year 2008, a lot of companies working on it, but it was such a, challenge here. The reason being agonist, all the synthetic agonists binds to a really hydrophobic pocket deep inside the protein. Maybe it's easier for me to explain it to you if I show you a real cloud EM structure of the TRPM1. As you can see, this is a cloud EM structure. This side is facing the cytosol, and the other side is facing the lysosome. In between, as you can see, blue represents the nitrogen, oxygen is red, meaning that they're very polar, very, water-soluble regions. But in between all these green is the carbon. So these are lipid bilayer regions, as you can see a ring goes through. This is where the natural ligand binds, but they're very polar molecules like PI352P. You could never really develop a brain-penetrable compound out of it. For synthetic molecules, though, the agonist, as you can see, they bind very deep inside here. So if you have a molecule, you need that molecule to be potent. It has to be really hydrophobic. But once it's hydrophobic, then it's metabolically labile. So very short half-life inside the body, but it's also really insoluble. So it's very hard to really formulate the molecule to get an optimal absorption too. So I'm gonna just go back to the slide set here. So for years, folks have synthesized probably more than, I don't know, like maybe 20 more patents now being published for the TRPM1 agonist. But none of them are really highly brain penetrable and potent at the same time. So we spent about 4 years working on it, and eventually we were able to actually get molecules with oral bioavailability bigger than 50%, with a brain to plasma ratio bigger than 1.5, which is extremely high already, and still have a very high potency. And the tools that we used, we started with the fundamentals. We actually obtained probably more than half a dozen cryo EM structures of our own with the synthetic agonist bound to it. And then we did a lot of modeling and synthesized more than 1,500 compounds of our own for about 3 years, and eventually get the desired molecules that are highly brain penetrable and potent. And for the next few slides, I'm going to just show you, that they did exactly what we're asking for. This is in vitro. You've seen that, activation of TFAT, seeing, extremely potent molecules, even at 3 nanomolars. TFAP gets translocated into the nucleus as well as TFC3. And if you're looking at induction of the autophagy, looking at the LC32 formation, even at 1 nanomolar, you're starting to see these bands, get intense. And now if you're looking at exocytosis, it's the same thing. You're looking at the marker for, LAMP1, which is usually on the lysosomal membrane, but after exocytosis, those molecules get fused with the plasma membrane, so you still see a ring formation over here. And then same thing happens here in vivo. And when we dose the rats with our lead molecule, as you can see, that these are downstream, genes that are regulated by the TFAP. You can see the increase of the LC3 mRNA increase, cathepsin B, GBA, as well as this is a sort of a binding molecules that binds to the GBA. And this is actually in the red whole blood, but this is actually looking at the brain, mouse cortex. And again, you're seeing LC3, both 1 and 2, it's increased as well. And finally, looking at the exocytosis, where you can see the activity of beta X and acid phosphatase, which is a lysosomal enzyme. Right? Usually they resides inside the lysosome, but once the exocytosis happen, then they get excreted into the plasma. So you're seeing the activity increase of those two enzymes too. And finally, I'm going to show you the in vivo mouse model. In this case, we're choosing 18 months old mice, which would be equal to like humans in 60 years old. So in 18 months old mice, their ALP pathways functions also decline. In this case, we inject the human alpha-synuclein fibrils to the substantial Niagara region of, basically a midbrain region of the mouse. And in another case, we actually supplemented with the CBE, which is a irreversible, GBA inhibitors. And that's basically messed up with the lysosomal function further. So therefore, you're seeing more severe phenotypes. And in both cases, we treat these mice with all compounds 30 mg per kg for 35 days. So you're looking at the first figure over here, that this is just body weight data showing that there is no toxicity for this compound. And now first figure over here, you're seeing the alpha-synuclein area in the dopaminergic neuron in the substantial Niagara region. So in normal mice, you're seeing 100%. But once you treat it with the alpha-synuclein, you're seeing increase of alpha-synuclein aggregates inside the neuron. With the double challenge, with the CBE, which is an inhibitor of GCase, you see even more buildup of alpha-synuclein in these neurons. And then when we treat it with our compound orally for 35 days, you're seeing the reduction of alpha-synuclein area dramatically all the way back to normal. And on the right side over here, you see the GFAP, and it's an indication of the information basically. And then with the treatment, these two red bars, you see increased inflammation dramatically. And again, treated with the drug, you see the two blue bars, they constant too. And you look at it further, looking at the neuron survival and the dopaminergic neuron, after the treatment, you're seeing significant decrease of the neuron. And with the double challenge, even lower, all the way down to like 70%. But once we treat it with our drug, you can see the Th positive neurons increase dramatically too. And finally, this is actually looking at the plasma level of the neurofilament L, which is an indication of the neuronal, death or neuronal damage, right? And again, you see with the double challenge, you see a lot much higher NFL in the plasma, but once treated with our drug, it really drops down dramatically too. So we feel very hopeful, and this drug could be really useful for both the idiopathic PD as well as the GBA positive PD. So at this stage, we spent quite a amount of time working on a number of other issues as well, and but we solve it all, including formulation, including all the biomarker discoveries on things in that nature. And we're very end of it now. We're finishing the 1 month GLP studies, so we're going to file the IND in the later in the fall, hopefully to start our phase 1 trials early January next year in healthy volunteers. And of course, this is a teamwork. We're a very small team, but we're highly experienced, and we're very virtual as well. So work with many teams, and we got a lot of support from a lot of experts, including Jonathan, who really gave us a huge lift in the 2 or 3 years ago. That really helped us a lot, and we really appreciate it. And I'm
Thank
not
you.
sure if I'm running out of time.
Yeah,
If
I think we're going
there's
to have to
any
move.
questions.
Yeah.
All right.
I think we're enough to move on. But
Okay.
thank you so much, Young Chen.
Sure. All right.
Um, we're moving on to Andres from Aspen Neuroscience. Can you share your screen there?
Hi, everybody. Can you guys see my screen?
Perfect.
Okay, well, first, I just wanted to thank Jonathan, Ben, and Vivian for the invitation, the opportunity to speak to you all. I'm really excited to present about Aspen. My name is Andres Leal. I'm a co-founder of Aspen Neuroscience and currently the Senior Vice President of Research. I started working on this project as a postdoc at Scripps Research Institute in 2012. So for me, this has been about a 13-year-long journey to get to a clinical trial where we're using autologous cells to replace lost neurons for Parkinson's disease. Aspen, so we started the company in 2018. Our lead product is an autologous dopaminergic replacement. We just completed our dosing in our phase 1/2A study, so I'll talk a little bit about that study, talk a little bit about the company, what our areas of expertise, and I finished kind of talking about how we think this could be applied to GBA, and I think that might actually be starting pretty soon, so we're really excited about that. Our lead indication is for sporadic Parkinson's disease, but we have been working on GBA for the last several years as well. We are really excited about other cells in the CNS, starting with a rare pediatric disease that could lead into broader indication. Induced pluripotent stem cells can become any cell type in the body, and we spent a lot of time, money, and effort creating a really robust platform to make high-quality iPSCs from anybody. So we're looking forward to being able to broaden the pipeline and the applications of the work that we've already done. So with an autologous strategy, no immune suppression is required. We believe that these cells will engraft and persist much longer and will have a better efficacy than allogeneic cells. Right now, there's some clinical trials that are reporting out for Parkinson's disease that use embryonic stem cells, or they use an allogeneic source for induced pluripotent stem cells. And those trials have the patients on immune suppression for 1 year, after which they're tapered off. But we don't really know how long is required or what the optimal immune suppression strategy is for a cell therapy for Parkinson's disease. But with an autologous cell product, we don't have to worry about immune suppression at all. We believe that there'll be enhanced synaptic connections and circuit reformation. It's scalable. We don't need very many cells per patient, so we don't have to expand these cells where there's potential for these cells to acquire mutations in culture. So Aspen is really built on 3 pillars of expertise. One is stem cell biology, and that's creating really high quality induced pluripotent stem cells for every patient. Unlike CAR T therapy, where you're starting with a bone marrow aspirate that could vary from patient to patient, we start with fibroblasts, we make iPSC, so we get a really consistent starting type from each patient. We don't change anything about our manufacturing platform from patient to patient. For us, it's really about choosing the right induced pluripotent stem cell colony that will differentiate in a predictable manner. So what enables us to move forward with an autologous cell is a bioinformatic expertise and the manufacturing expertise. So we use a bioinformatic-based strategy for quality control. This is something that's scalable, replaces animal study. We look at bulk RNA sequencing to really predict the cell's ability to make dopamine, to engraft, and to innervate once transplanted in vivo. So we are really, because manufacturing and the quality of the cells is so important, we made our own manufacturing GMP space. And here we've already made 100 different GMP iPSC lines that passed our release criteria. We've completed the manufacturing and the dosing for the subjects in our first 2 cohorts of our phase 1/2A clinical trial. We've developed a thaw and inject formulation, which we think is really a game changer. Now the cells can be frozen in the OR, they're thawed, put into a cannula and directly injected. They don't require processing in a cell culture hood where you need to thaw the cells, concentrate them, count them, remove the cryopreservative. So this is something that I think will really enable us to expand beyond the medical centers which have available cell culture space near the OR. We have been focused for the last couple of years on automating the system. So we're using robots to do the reprogramming process. developing software that can autonomously run the reprogramming process, remove the technician part of the process where somebody actually has to scrape the cells or pick the colonies or choose which cells to expand. And really excited about this and implementing this in our future clinical trials. So we start with skin cells. We make induced pluripotent stem cells. From there, we make dopaminergic neuron precursor cells, which are then transplanted back into the same patients, directly into the putamen, where most of the A9 dopaminergic neuron innervation is lost in Parkinson's disease. Along the way, we do whole genome sequencing at every stage to make sure that we haven't selected for any cells that have acquired mutations. And then we do bulk RNA-seq as well. So both of these methods ensure that we have cells that are going to be safe and efficacious. So our preclinical package, our IND, was submitted 2023. It was approved in 2023. We worked with Parkinson's donor cells in our preclinical development process. Put cells from different donors into rats and showed that they can restore Parkinson's. The model, they can restore dopamine signaling in a denervated rat brain. So we get really nice grafts that survived for 9 months and look beautiful in our rats from Parkinson's patients. We did a trial-ready cohort, which I think is pretty innovative and exciting for those that are interested in kind of clinical trial design for cell therapy. Prior to IND approval, we had already been making or recruited patients and made their cells so that once the IND was approved, we were ready to go. This is a dose escalation study. We have 2 different cohorts of different dosages, and we're going to be looking at up to 18 months. Looking at on time without troublesome dyskinesia, off time, the UPDRS motor score, activities of daily living. We're looking at PET scans every 6 months, as well as using some digital health technologies as an exploratory endpoint. So our patients are offered the Roon watch and the Apple Watch with the Roon Strive application. And also a wall-mounted device that measure gait, speed, REM sleep, dyskinesia, other things like that we can use as an exploratory endpoint to investigate the effects of our cells. So this is an MRI from our very first patient. This is done under intraoperative MRI. We go through the back of the head, and this allows us to lay down the cells along the long axis of the putamen. The cells are admixed with gatoteridol, which is an MRI contrast agent. So you can see immediately after the surgery that we've placed the cells exactly where we want them to be. This gives us sub-millimeter accuracy. So we get confirmation that there's no brain bleed, that we can avoid blood vessels. The cells are deposited accurately every single time. That's something that we're really excited about as well. So from our clinical experience, we've dosed all our patients in the first 2 cohorts in 2024. No surgical adverse events were observed. We were able to accurately and safely place the cells. We're really excited about the efficacy that we're seeing right now. And starting next month at ISCT and then again in ISSCR in June, we'll be presenting the very first efficacy data from this. Other groups have started to present their efficacy data from allogeneic cells. So we're very excited to present our clinical efficacy data. I hope to come back to another foundation meeting in the future and to be able to share that with you all. But specifically of interest to this group, we have done some work supported by the Silverstein Foundation on GBA dopaminergic neurons. And we started out with a hypothesis that we would have to correct the GBA mutation to make these cells appropriate for an autologous cell therapy. And so we took some GBA E326K patient cells, N370S, 2 different donors, and we used CRISPR to correct the mutation. We turned it from a heterozygous mutation into a homozygous mutation. We completely knocked out the mutated allele, and then we made dopaminergic neurons from all these different isogenic cell lines. And what we found was, even though there was a decrease in GCase activity in the mutated cells that were further decreased when we made it homozygous or we knocked it out, we didn't actually see any change in lysosomal function, mitochondrial function, resilience to alpha-synuclein fibril treatment. And so I think this is really because Parkinson's is a late onset disorder. It's not a congenital disorder where there's a problem with the the development of these cells. And the reprogramming process elongates telomeres. It completely eliminates the epigenetic markers of aging. It restores mitochondrial and lysosomal function. So the cells we're making really are fetal-like cells which have restored function. And so the entire couple of years that we worked on this, kind of thinking about a gene-edited product, convinced us that we actually don't need to do gene editing for these cells for these patients. We took our cell lines and we made, we made 14 different lots that were clinical grade for these cells, and they passed all of our release criteria. They released dopamine similar to our idiopathic cells. They passed all of our flow release assays. So right now we're working towards building the case that we could treat patients with GBA Parkinson's with their own unedited dopaminergic neurons derived from induced pluripotent stem cells. We're having KOL meetings, talking to clinicians, and excited about potentially moving this forward into the clinic. So with that, I think that was my last slide. Quick introduction of Aspen. I'm really happy to answer any questions that anybody might have. Thanks for the time and the attention.
If you have a question, please put it in the chat box.
Can I ask a question?
Yeah, you have a question? Sure.
Yeah. So if there is existing neuroinflammation, do you know what the impact would be on the transplanted cells?
Yeah, so I think what we've learned from fetal cell work that was started in the 80s and 90s, where they took aborted fetal tissue and they would dissect out the ventral mesencephalon to treat Parkinson's patients, is that even in a Parkinsonian brain, these cells can survive for up to 24 years, can give graft-related benefit to these patients for over 10 years. Some of these patients were able to come off of their L-DOPA medication completely. And in some of these patients with severe alpha-synuclein pathology, some of the grafted cells started to show alpha-synuclein pathology as well. And so it increased over time, but even after 10 years, it was a single-digit percentage of these cells, and it didn't appear to affect the function. So I think if these cells, these fetal cells, can survive in that environment, we believe our iPSC cells can do so as well and give, long-term benefit to these patients, maybe over 10 years of benefit to these patients. Right now we have 1-year data that we're really excited about. And this is the experiment that we have to do, but we believe that the preclinical evidence or the clinical evidence from the fetal cell trial supports the long-term efficacy of these cells.
Quick question. Andres, hi.
Ben.
I'm wondering, how far are we from being able to automate the entire manufacturing process?
Right now, the technology we've been working on automates 90% of the process. So it automates the reprogramming, the differentiation, the fill finish. And so we're going to implement that, and the plan is to implement that into our pivotal trial. So we want to be ready with that within the next couple of years, maybe even sooner. Right now it works. It's transferring it to GMP, and that's where we are now. So it'll be soon.
Thank you very much. There's one question in the chat from Dan. I can read it right now. Regarding the clinical study, how do you determine the inclusion criteria? And are there certain patients that would benefit most?
Yeah, so our inclusion criteria is similar to DBS. So right now for idiopathic program, patients with mutations that are associated with causative for Parkinson's or associated with Parkinson's, likely pathogenic in ClinVar, are excluded from our trial. We're kind of rethinking that in a separate cohort and trying to get some data to see if that's the right exclusion criteria or not. But I really believe that the patients that are likely to be the best responders in this type of therapy are patients that respond well to L-DOPA, that have now begun to have some troublesome L-DOPA-induced dyskinesias. And so this is really a dopamine replacement therapy, but it's done locally and it's released in a physiological way. So we believe the patients that responded well to L-DOPA will respond well to the therapy.
Okay, I think we need to move on, but there's some other questions in the chat as well. If you're able to answer them, feel free to go ahead and try, Andres. Thank you, though. That was a great presentation.
So do I have more time? Would you guys like? I see the ones in the chat. Do you want me to keep going?
I think we need to move on to the next top.
Okay,
I'm
yep.
not
Thank you,
sure.
everybody.
Thanks.
All right. So next up is Lorraine Kalia from University of Toronto. Lorraine, can you share your screen? Great, thank you.
Great, thank you. Thanks, Jonathan, for the invitation. And I'll just disclaim up front that I'm not from industry. I'm an academic clinician scientist at the University of Toronto in Canada. And I thank Jonathan for giving me the opportunity to take the next 10 minutes just to present some of the work that we've been doing collaboratively in Toronto using this technology of low intensity MR-guided focused ultrasound for drug delivery. And in the Parkinson's disease space, basically, the test case has been in a GBA-related Parkinson's disease, seeing if we can actually deliver glucose-free residase using this technology. So for this audience, I think I don't really need to review, but I do just want to emphasize that in Parkinson's disease, GCase deficiency, which you already saw some evidence for, is thought to be an important pathogenic mechanism, both in GBA-related Parkinson's disease and a lot of preclinical work. Some here summarized from some of the pivotal work done by Dimitri Kranz's lab, showing an interaction between GCase and alpha-synuclein and the way that alpha-synuclein is processed, including alpha-synuclein aggregates in cells, is mediated by glucose reversidase. And then evidence in actual human brains from work done by Shapiro's group, looking at postmortem tissue, demonstrating that actually GCase activity is reduced in GBA-related Parkinson's disease, shown here in the white bars, but also in cases of sporadic Parkinson's disease, thus providing some evidence that this is a mechanism that's going to be relevant to both GBA-related Parkinson's disease and at least a subgroup of probably people with sporadic Parkinson's disease. So one could, quite simply say, well, if GCase is deficient in Parkinson's disease, then isn't it just something that we could replace? And this, of course, is a logical proposition. And one could even ask, well, is there not recombinant GCase already available? And indeed, that is already a therapeutic product. As everyone on the call is probably aware, Gaucher disease is an autosomal recessive condition that is a consequence of glucose reversidase deficiency. And many decades ago, products were developed in which glucose reversidase was generated recombinantly. It's delivered intravenously to manage the systemic complications of Gaucher disease. And there are 3 products that have been given to many, many people around the globe. And this is actually standard of care for people who have symptomatic Gaucher disease. However, intravenous delivery of glucose reversidase is not an approach that we can simply adopt in Parkinson's disease because this is an enzyme that does not cross the blood-brain barrier. And here's just a couple of pieces of evidence for this. This is a very early PET study where glucose reversidase, the original product Cerezyme, was radio-labeled and given to individuals, either healthy subjects or people with Gaucher disease. And you can see the bright signal indicates great uptake of within organs, within the spleen and the liver. Up here's the head, and you can see very little brightness there. And then just experimentally, if you take glucose reversidase and give it intravenously into rodents, these are just Western blots. This is the positive control showing glucose reversidase. On the Western blot, animals that are intravenously delivered glucose reversidase and then brain tissues looked at, you can see there is no evidence of any glucose reversidase there. And that, of course, is not surprising. We all know that the blood-brain barrier exists for protecting the brain and is both a passive barrier to products within the lumen of blood vessels, and also has an active component to either actively transport things across the blood-brain barrier or even actively pump things out across the blood-brain barrier. And glucose reversidase, being a protein or an enzyme, is one of these products that typically does not cross the blood-brain barrier. There are other things that are interesting from a therapeutics point of view in Parkinson's disease, some of which you already heard about. So stem cells, for instance, are something that are being very actively investigated in Parkinson's disease, but if injected intravenously, don't typically cross a blood-brain barrier. Some of the larger small molecules, and then these kind of nucleotide type therapeutics that are very prolific right now in the neurodegenerative space also can't cross the blood-brain barrier if you give it that way. And so, of course, there are a number of different technologies that allow us to get things into the brain. This is, I thought, a nice summary of some of the invasive technologies that are more traditionally used. So direct brain injection, like you just heard about with the stem cells, intrathecal delivery, which is typically being done with viruses right now, as well as with ASOs. But there are a lot of more non-invasive or less invasive technologies that I think are quite exciting and will open up a lot of opportunities for drug delivery in Parkinson's disease and GBA-related Parkinson's disease, some of which include the neurotropic viruses, which I think you'll probably hear about more today as well. And I'm just going to focus on this technology of focused ultrasound, low-intensity focused ultrasound with microbubbles. If any of you followed focused ultrasound literature in the area of Parkinson's disease and movement disorders in general, you're going to have heard primarily about high-intensity focused ultrasound. This is a technique that is used to actually make an ablation, so to actually cause a lesion within the brain using high-intensity focused ultrasound. And focused ultrasound itself has been a technology that's been around for a long time, but one of the reasons why it's had a bit of a renaissance and a resurgence is because of its partnering with imaging. So this is an MRI machine. This is an individual with a helmet on. And basically what's happening underneath this helmet, there's a number of ultrasound targeting a very deep area within the brain. And with the capabilities of the targeting together with the visualization of the MR, you can actually then look at the specific areas where you want to, in this case, lesion the brain, and in our case, actually deliver medication. Another indication that's emerging in the literature is the use of low-intensity focused ultrasound for neuromodulation. So the intention here is not to cause any damage to the brain, but to alter the activity of neurons. And by being able to do this very strategically in specific places, one could imagine modulating a node or a circuit within the brain, and that's being investigated, similar to transcranial magnetic stimulation, and many of the studies are being done to see if you can replicate that kind of activity using focused ultrasound instead of magnetic stimulation. And what we've been using is low-intensity focused ultrasound, again, a frequency at which damage to the brain does not occur. But what it's combined with is intravenous delivery of microbubbles, which are just shown here. So this here, again, is the blood-brain barrier. This is a vessel. This is a microbubble within the vessel. And what happens when a microbubble, so these are the same kind of microbubbles that are delivered when somebody's doing an echocardiogram to look for a PFO. And so these are clinically used. That the mechanical movement, the expansion and contraction of these microbubbles, as well as the movement of the microbubbles, causes actual mechanical disruption of the blood-brain barrier, including the basement membrane and the adhesion between cells, temporarily to allow what's within the lumen to actually escape into the actual parenchyma of the brain. So this, like I mentioned, is a very collaborative project. At the Toronto Western Hospital, we provide the expertise around Parkinson's disease. And at Sunnybrook Hospital, which is in the north part of our city, we have experts in the area of focused ultrasound, including Nir Lipsman, who's the clinical expert, and Claire Heinonen, who's the physicist behind much of the development of this technology. And Nir and Clairvaux, early on with their collaborators up at Sunnybrook, had been looking in Alzheimer's disease as well as ALS with proof of principle studies in humans to demonstrate that this technology could be used safely. And so in Alzheimer's disease, this is an Alzheimer's disease brain. You'll see a couple of these different kinds of scans where the surrogates to indicate that there has been a breach of the blood-brain barrier is co-administration of gadolinium, which normally, so in this case, blood-brain barrier disruption, you can see there's no brightness in the brain. The area of the brain that was targeted with focused ultrasound was right here. And then when a repeat scan is done with gadolinium, again, you can see that there is a brightness being breach of the blood-brain barrier. And then 24 hours or 48 hours later, when the scan is done again, you can see there's no more gadolinium in this area, indicating that the gadolinium can't cross the blood-brain barrier. So this was first done in Alzheimer's disease, done on eloquent cortex, so over the motor cortex in a person with, in a handful of patients with ALS. And then colleagues as well in Spain. So Jose Obeso's group has been looking at this in Parkinson's disease, and they did the early studies of just opening up the blood-brain barrier in patients with Parkinson's disease dementia within the cortex as well as within the striatum. So we came together in a collaboration, the Sunnybrook team and the Toronto Western team, to actually start to deliver something, not just open up the blood-brain barrier, and building on the safety data that I just showed from those 3 other trials, to see if we could actually deliver something. And while we contemplated a variety of strategies or a variety of agents to actually use as something to deliver, glucose riboside made the most sense for a variety of reasons. One, it's known safety when delivered intravenously already, because patients with Gaucher disease have been given it for decades. Secondly, its availability. We can actually buy it off the shelf because it's a commercial product. And thirdly, because of the background in terms of the biology of glucose riboside deficiency actually being a cause of, or at least a contributor to, the neurodegeneration of Parkinson's disease. So we embarked on a very small clinical trial published here, a phase 1 trial where we intentionally recruited people with GBA related Parkinson's disease. The goal here was really to look at safety. We quite easily recruited 4 individuals. Their genotypes are shown here. You can see that one also had a LRRK2 mutation, and our 4th person here actually had 2 mutations, so had Gaucher disease. These, again, are the MRI scans just showing the gadolinium enhancement in each of the 4 after opening up of the blood-brain barrier. This is one patient in a variety of different views, and you can see the brightness of the gadolinium demonstrates that we did open up the blood-brain barrier using this focused ultrasound. From a primary outcomes point of view, our focus was really safety and feasibility. We had no serious adverse events. The adverse events that we did have included mostly ones related to the frame that needed to be put on place. And you can see this is one of our participants here. This is the frame. It has to actually get pinned into the scalp. And so this comes with complications in regular scenarios. Interestingly, the technology from the hardware point of view around focused ultrasound is advancing substantially, moving towards probably in very short term a frameless technology. You can see that this patient had her head shaved, which was required for the penetration of the ultrasound. Our second study, which is ongoing right now, is an example of how this technology evolves, that the head shave is no longer required. And like I mentioned, there's an anticipation that this is all going to go frameless relatively soon as well. Dyskinesia was something that we observed in some patients. And so this is actually the same patient that you just saw in the prior scan. This is actually post one of her procedures. She had a little bit of dyskinesia originally before being in the study, and after procedure, you can see that her dyskinesias were increased. We think that this is related to opening of the blood-brain barrier and increase of her dopaminergic medications getting in. These resolved over time. And the other adverse event was a T2 signal change. So we kept very close eye on T2 signal change as indication of potential microhemorrhage, because this is a potential complication of focused ultrasound. And we saw these signals that did not represent a microhemorrhage as they resolved over time. And then there was also this T2 flare signal, which we're not quite sure what it represents, but again, it was a reversible signal that was not associated with anything clinically. Exploratory outcome measures were really just very exploratory, and in this very small number, very little for us to be able to conclude from. We looked at FDG PET, which interestingly seemed to decrease in treated patients. And this was interesting in part because often you expect an increase in activity in Parkinson's disease. We looked at clinical scores, but also changed treatments as we needed. And so the value of what these actually tell us is very limited. As you mentioned, we made some changes to dopaminergic medications. We did a couple of different measures as well, and nothing really that we can conclude anything from. Last piece I'll just mention, what we did not have in this study was a direct indicator of glucose cerebrosidase being delivered. We had this indirect measure of gadolinium being administered after the fact, but we did not have actual ways to demonstrate to ourselves that glucose cerebrosidase was in the brain. There are direct indicators from other studies that suggest using these kinds of parameters will provide penetration of glucose cerebrosidase into the brain. And this is just an example, again, from our colleagues, Jose Obeso's group in Spain. This is a primate study where they intravenously delivered an AAV that expressed GFP. You can see this is a primate brain with the same kind of gadolinium enhancement that they used as a surrogate. And then when they actually were able to do the postmortem on this this monkey, that there was actually GFP signal within the neurons. And in another disease population at Sunnybrook Hospital, this is looking at actual human data for individuals with metastatic breast cancer being delivered an anti-HER2 antibody, so trastuzumab, which was radio-labeled. This antibody is actually larger in size compared to glucose cerebrosidase, but the capability of being able to use a radio-labeled antibody allowed for detecting whether or not there was increase in signal, which there indeed was in patients that were examined after focused ultrasound. So again, a direct measure for this study, but suggestive that if you had an antibody, that's far larger than glucose cerebrosidase, that's using similar parameters, you'll get glucose cerebrosidase in there. And so we're currently in the process of doing a larger trial. We are recruiting, again, GBA-related Parkinson's disease, but also sporadic Parkinson's disease patients. The difference here is we have been, instead of unilaterally delivering glucose cerebrosidase, we have been attempting to do a bilateral delivery simultaneously at the same time, and trying to see if, from the point of view of safety and tolerance, that, as well as adding on some additional measures to hopefully be able to look at CSF at least to be able to look for penetration of glucose cerebrosidase. And I'll just add in the last last sentence on my part is that, of course, there is a ton of preclinical work that still needs to be done around this. This is a project where we started in humans and we'll need to work backwards to laboratory models, having to understand not only does glucose cerebrosidase get past the blood-brain barrier, but once it gets past the blood-brain barrier, actually, where does it go? So with that, I just want to thank funders, including the Silverstein Foundation, which provided funds for this, as well as the Weston Family Foundation, which provided a lot of funds for the current clinical trial. As I mentioned, this is a very collaborative effort across Toronto sites, including the group at Sunnybrook Hospital, as well as the Toronto Western Hospital. And also thank the patients who have been really tremendous and brave to be part of this study. So thanks very much.
Thanks, Lorraine. Jeremy's up next, but Jeremy, why don't you share your screen? And I think there was a question in the box, chat box.
Yes, Shalini, we do have CSF before and after. If you have assays that you have that we could do on the CSF before and after, that would be wonderful. We've tried to do mass spec. We've done a little bit of GK's activity with one collaborator, but their assay was not particularly reproducible.
Okay, thanks. Jeremy, you're up.
Great. Can you see my screen?
Perfectly.
Great. All right, so thank you very much. for the invitation. So my name is Jeremy Spashwan. I'm an associate director here at Congruence, and we're really excited to show you some of the work we've been doing over the last 3 years. So I don't know if I was following the criteria too closely, but we were asked to provide a personal background. So this is kind of how I got into the industry. So I'll briefly go over my scientific career. So I completed my PhD at McGill University. So at the time I was working on the inflammasome and gastrointestinal homeostasis.
Jeremy, you're in
And
presenter mode, just a warning.
Oh, okay. Just a sec. Thank you, Stephanie. So what are you seeing right now?
Just you're the like this. You're in just the screen without being in full view. Like we can see all your slides.
Oh, okay. Let me stop sharing. Sorry about that. This one. Is this better? Or no?
We can't see anything now.
You can't see anything now. Okay. This is good?
Yes, now it's good.
Perfect.
Perfect. Thank you.
Great, thank you. All right, so from there, I moved to enGene Inc. So enGene is a non-viral gene therapy company currently in clinical stages. So there I worked as a scientist at various levels. So initially, I was hired to develop colitis models based on my background, and eventually moved to a more in vitro assay development side of things. And near the end there, I was the biology lead for their discovery group to expand on different mucosal tissues. And since 2002, I've been at Congruence as the bio lead on the GBA-PD program. And as in all small biotechs, you wear multiple hats. So this is a great opportunity for me. So what do we do at Congruence? So our mission is to encode protein motion to unlock a new era of transformative medicine. So our technology focuses on a powerful computational drug design engine called Revenir, which captures the biophysical features of proteins and their full conformational landscape. So we use Revenir as a way to identify, first of all, pockets where small molecules can bind, and also how these different molecules can then revert the protein to a more of a wild type like feature. So the goal is to develop pharmacological correctors and activators, and through rational design. And we focus most of our activities on diseases which have either activity defects or misfolded proteins. So our pipeline is across various genetic diseases, as mentioned, focusing mostly on protein folding. So we are kind of disease agnostic. And as you know, one such disease is, or one protein which is typically misfolded is GCase. So for Gaucher disease as well as for GBA Parkinson's disease. So our approach is a two-pronged approach. So internally, we have two screening pathways. One which focuses on the loss of function that's observed in GCase, which leads to the disruption of lipid homeostasis and impaired alpha-synuclein clearance. And so at the bottom here, you have an example of how we use Revenir. So in green is the state when GCase is activated compared to an inactive state, which is in red. And then we can look at how binding of our small molecules can put this GCase in a more of a activated format. And secondly, we have another screening platform which focuses more on gain of toxic function from GCase, which is mostly related to protein misfolding and retention in the ER, which can cause ER stress and mitochondrial dysfunction and neuroinflammation. And so in the GCase corrector side of things, here we're showcasing an effect with alpha-4p, where you see that conformationally there are quite different. There are big differences between wild type and alpha-4p, and how when alpha-4p is then coupled with a compound, it reverts back to a more of a wild type like conformation. So for today's presentation, I'll focus on our screen that was focused on the corrector series. All right, so we started with a very simple question. So can we use GCase glycosylation as a surrogate marker for lysosomal trafficking? So GCase, as is being synthesized, is glycosylated at 4 different sites, and then traffic from the Golgi to the lysosome, right? And as you know, glycosylation is quite a complex pathway where different sugar moieties are added and removed, and which leads to a mature glycosylation. And what's specific about mature and glycosylation in the case of GCase is that it's resistant to this enzyme called PNGase, and we can use PNGase F as a way to remove all glycosylation on GCase. So can we screen using this simple approach? So first we use just normal cells. So these are wild type. So you see that when they're untreated, when the lysates are untreated, you have a smearing of GCase due to glycosylation. When you treat your lysates with endo-H, you have these 2 fractions. And for these cell types, there's very little that's remained in the ER. And when you remove all glycosyl moieties, you see that in this case you have the predicted height for GCase. Now, if we use mutant cells, so GBAs that are homozygous for N37S, and you do an endo-H treatment, you see that in this case there's barely any of these what we call mature GCase. And if we use IFG, or isophagamine, which is an inhibitory chaperone, you can see that we can increase this endo-H resistant fraction in a dose-dependent manner. So using this simple approach, we decided to run a screen. So we did a in silico screen where we screen millions of compounds for binding to our pharmacophores on GCase. And from there, we ordered compounds and we tested them in the lab. And out of a screen of about 250 compounds, we had 10 compounds which were 3 standard deviations away from the mean. And here I'm just highlighting one, which we'll call compound 1 for the purpose of this presentation, which quite significantly increased the endo-H resistant GCase. And then when we did a dose response, we saw that as we increase incubation with our compound, we could get a dose-dependent effect. And so we identified a diverse set of molecules that could be then further characterized for their GCase activity profile. So what we've been using at Congruence is a PFP-FD glue substrate, since this substrate is specific for GCase once it's a lysosome. So we'll incubate our cells with different compounds for hours or days. And what you expect is as you have our chaperone or a corrector, you'll increase your total amount of GCase in the lysosome, which would then lead to an increased fluorescent substrate using a plate reader. And so from there, when we looked at the potential of compound 1 to increase GCase activity, we could see that we had a mild effect and only at high concentration. But after some medicinal chemistry, we identified a second compound in the same series, which could quite significantly increase GCase activity in our RPE cell lines. And interestingly, this effect was also maintained in typical heterozygous mutation, including alpha-4p and E326K. And what's most importantly here is that we still maintain the endo-H resistant effect. So we have a dual pharmacology effect with this compound, right? We can not only increase levels of lysosomal fraction, but we can also activate GCase in the lysosome. And so from there, we wanted to really try to increase our cycle time. So performing the EndoH assay is quite time consuming. And when you talk with your chemistry fellows, you'll see that the first thing they want to do is accelerate how fast you can go from, to design your small molecule, to make it, and then to test it. So the faster you can go through this cycle, the faster you can generate more and more compound and more matter to work with. So to do this, we used a fairly simple approach where we CRISPR'd in HiBit on GBA1. So HiBit is a technology from Promega. It's a complementation assay that when the HiBit tag is coupled with a large bit, it emits light, which is a very specific and very sensitive approach. So if we create these GBA1 HiBit cell lines, can we still pick up an increase in total GCase activity? And then on the right-hand side, what you see here is the luminescence, which we see that in a dose-dependent fashion, we can increase the total levels of GCase. So from there, the program is very well positioned to screen the next generation of correctors to accelerate our drug discovery process. So this is all I have for today. So I'll say thank you, and I'm happy to take questions.
No questions, I guess. Vanqua is up next. Kevin or Jim, I'm not sure which one of you is presenting.
It'll be me today.
Okay, great.
Okay.
Share your screen.
You betcha. Is that full screen for everybody?
No, there it is. Now it is.
Okay, great. Thanks. It's great to be here on behalf of Vanqua Bio. And I think the most important thing I would like to hear for you to hear today, besides the science, is the importance of relationships. And what Jonathan has done is create a cohort of the next generation of GBA1 scientific leaders. And so as you learn and are competitive against one another, make sure you lean in on those relationships so you can build companies and you can build drugs. The reason we're Vanqua today is very much because of that. I was a consultant, and before there was a Vanqua, there was a Jonathan who had us look at an opportunity down at the Feinberg School of Medicine at Chicago. And in so doing, I got to know some important team members. Jim ended up partnering with Dimitri Krantz to co-found Vanqua. I stopped being a consultant and stepped into as a CSO. And because of these relationships, we were able to bring in really key people, including Diane Asselstein, who leads our biology, from Dimitri's group, and build out what we need to do to make this molecule. My background is I'm a drug hunter. I know very little about Parkinson's before this company, but I really, really have learned a ton because of the right people around me. And so we went through and we made a molecule because of our combined experiences. I was at Array Biopharma, where we moved 20 molecules into the clinic. Many are approved drugs in my 13 years there. Since then, I've been in other collaborations, including leading science at Calico, where I got to know Jim, which led to a trusting relationship that helped us get this started. And since then, I've been part of other biotechs to make important drugs for Duchenne's muscular dystrophy and cardiovascular disorders. So with that, we'll get into it. We're here today because of GBA1 being a primary risk factor for Parkinson's disease. These mutations lead to about 30% less activity. This decreased activity leads to dysfunctional lysosomes, loss of lipid homeostasis, and the hallmark of Parkinson's disease, which is insoluble alpha-synuclein. The Vanqua therapeutic hypothesis is quite simple, very similar to others, is we need more effective GCase. And if we're able to just activate the wild type allele, which all patients with GBA1 have, is we can take and activate this GCase, say to 50%. And with 50% activation, we could restore normal levels of GCase activity compared to wild type levels in patients with GBA-PD, which would lead to functional lysosomes, restore homeostasis in the lysosome, and reduce alpha-synuclein. Now, how did we do this? So I added this slide because I think it's important. What was the process here? Did we inherit a molecule or did we go find a molecule? What we had to do before we made the molecule was make the right assays. We had to build research tools. You have to build relevant research tools, but you have to build research tools where you can ask questions quickly that tell you enough truth to predict what will work in the more challenging cell types. We started out, as a drug hunter, I like to make molecules with the right profile. Here, we need to be fully CNS penetrant. CNS penetrance matters because the more you're off of unity, the more you have a peripheral burden, which could lead to off-target tox effects in the liver, the kidney, anywhere that's not the brain. So make sure you're fully CNS penetrant with your molecules if you can be. So we started out with that design. And the other key element to our approach has been ensuring that we measure what matters in the cell types that matter, and that's lysosomal GCase activation in neurons and showing that we don't accumulate alpha-synuclein nearly as quickly with our GCase activators. Our iPSC systems, which are day 50 and day 80, which are patient-derived, accumulate threefold accumulation of alpha-synuclein between day 50 and day 80. This allows us to measure effects of our molecules without any exogenous insult. We're basically recapitulating the disease in the dish. We were then able to find other assays that would predict what molecules would do well in this assay such that we could go fast and have a cohort of molecules that went into these longer-term neuronal assays that take 28 days from day 50 to day 80 or day 78. We measure GCase where it matters, which is in the lysosome. This is very, very important. GCase is a lysosomal assay. If you're measuring a lysate, you can be misleading yourself. So whether we're in vitro or in vivo, we measure GCase using probes that are specific to the lysosome. You can spike this into blood from patients. We're going to show you healthy volunteer data today. This probe goes selectively into the lysosome and then is selectively hydrolyzed by GCase, as others have shared, in the lysosome. And you get a fluorescent signal, and our molecules dose-dependently increase this fluorescent signal to show us what we're doing in vivo. In cellular systems, on the left-hand side, you see we have a nice concentration-dependent activation of wild-type GCase. This is where we're going to get most of the bang for our buck with this molecule. We have an AC50 or 50% activation with a concentration of 300 nanomolar. On the right-hand side, we show that we also have good potent activation of the most common mutations within GBA-PD, which make up about 85% of GBA-PD. And again, these are lysosomal assays in which we see between a 400 nanomolar and 1 micromolar activation of these key mutants. So we consider this bonus activity that we get on top of the wild-type allele. Moving to the cell type that matters, which is the neuron. Again, these are patient-derived neurons that are differentiated, and we start using these around day 50 when they're mature. We potently activate GCase in these neurons with 50% activation or AC50 or 300 nanomolar. And when you go to the right-hand side, if we start dosing 101 and other analogs at day 28 and look over day 78, we show concentration-dependent blockade of alpha-synuclein. And with the 50% activation or AC50, we see more than 70% blockade of alpha-synuclein. This allows us to put together with our therapeutic hypothesis, where do we want to engage GCase in the clinic? We feel if we have 50% activation at the C-min, we're going to have a significant effect for patients. Eventually, when we run the longer term trials, and then of course during the day will be a C-maxes that lead to with higher activation. We're able to use our same tool that we showed before to look at in vivo target engagement across species and now human. I show some data in the left-hand side where after a 1 mg per kg dose, we see 50% activation and up to almost 250% activation with 10 mg per kg. And what we do in the periphery, we do in the CNS. This molecule is fully CNS penetrant, whether you're looking at total drug, or more importantly, unbound drug, we have a KUPP of unity across species. Here we show that 10 mg per kg readily covers the mouse AC50 in both compartments. We are now in the midst of a 1A, 1B study where we completed the 1A in healthy volunteers and are moving forward with cohorts for GBA-PD and IPD. We'll show data from the MAD in healthy volunteers where we wanted to understand the safety and tolerability of this molecule, as well as make sure that we had pharmacokinetics consistent with once-a-day dosing and target engagement specifically for GCase. in GBA-PD and IPD patients. We are moving forward. We have completed some work in our 28-day study with 101 and for 2 doses. And we are now in open label extensions for up to 2 months where patients with GBA-PD or IPD can be dosed for a total of 3 months. And we'll report these data out later this summer. Besides safety, tolerability, we're also looking at sphingolipid regulation, as well as a variety of markers, including digital biomarkers. This is a great profile. The molecule is very safe. We've not had any serious adverse events, and we've not had any dropouts due to adverse events to date. And we just recently unblinded the safety, the healthy volunteer data, and there's one clear line here. When you look at the incidence in placebo, it's at 85%, which is higher than your active drug, which is consistent with a safe profile. So we're pleased with what we're seeing, and we're moving forward in the extended 1B studies with plans to go to pivotal trials in the coming years. In terms of pharmacokinetics, this molecule gets on board well. On the left-hand side, we show in the SAD study, or single ascending dose study, we see dose-dependent increases in concentration up to 600 milligrams, and then some loss thereafter. And importantly, on the right-hand side, in the MAD portion, or multiple ascending dose portion of this study, we see dose-dependent increases in concentration from 150 to 450, all the way through 450. So we show 50, 150, and 450, and then dose saturation, which you often see with molecules at higher doses at 600 milligrams. There are a few advantages to this molecule. It's a long half-life drug, about 100 hours, and this gives you accumulation factor, so you get quite a bit of drug on board. And once at steady state, this molecule has a minimal Cmin, Cmax variation, meaning you can readily cover your target without having excursions to high Cmaxes, which might be affect off-target anti-targets. So this is consistent, again, with our favorable tolerability profile to date. And moving to the CNS, which is the compartment that matters. And similar to what we saw in preclinical species, DQ101 is fully CNS penetrant, regardless of dose or duration of dosing, where we look at the CSF versus unbound plasma ratio, where we're at unity or greater, regardless of the cohorts we look at. And we see a very attractive plasma concentration versus CSF concentration over time. And again, this means you can hit your target without a peripheral burden, which could lead to untoward side effects. Now, for target engagement, that we couldn't be more pleased with these data. We wanted to get to an AC50 at Cmin. This measurement is taken on the last day of dosing at 24 hours. So this is the Cmin. This is the minimal activation we'd expect that we'd see over time. And so after the 24-hour time point, we show that 50 milligrams gives us modest activation. And with 150 milligrams, we're hitting that key 50% activation, or AC50, with the mean and median across this cohort. So we're very excited with 150. And if we dose up at 450, we're seeing 100% activation in these healthy volunteers. 600 was Noisier in PK, and it's Noisier in PD as well, but certainly sustained activation over 14 days has us very excited about what we're going to do in patients over the coming months. So what we know about GBA-PD is that patients have less active GCase. We recapitulated these data in a phase 0 study, a non-interventional study. And what we've shown you today is that we can restore the activation by 50% in healthy volunteers. And what this would do in GBA-PD patients would bring their GCase activity back to that of wild-type controls, or even greater. We couple that peripheral data with full CNS penetration, meaning what's happening in the periphery will be happening in the CNS. And we now can couple that AC50 that we're showing in the clinic to an AC50 that blocks alpha-synuclein in patient-derived neurons, which recapitulates the disease in a dish. So we're excited. GCase clearly is genetically validated. That's why we're here today. And 101 is the first molecule to show sustained 75% or greater lysosomal GCase activation in humans, and it blocks the accumulation of alpha-syn in the model that matters. This is clearly a once-a-day dosed drug. We have robust CNS exposure and good safety data, and we will continue to collect that over the coming months. And the team has put together a phenomenal design to allow us to understand how we're going to help patients with GBA-PD and the potential perhaps for IPD as well. So I'll take any questions.
Can I ask, Jonathan?
Yes, please.
Hey, Kevin, this is Mander. Really beautiful talk. I'm really wondering if with this data, you put kind of an end of that long argument that GBA mutations might be gain of function, which I myself never thought is true, but this is very clear here. You are activating, and what happens actually, you ameliorate disease phenotypes and biomarkers as well. Do you have any comments on that, Kevin?
Well, that's our belief system. It's, before we started dosing patients, we'd done a lot of, well, Dan had done a lot of work in the neurons to show that this mechanism matters. And so moving forward into, we'll be reporting out, effects across IPD lines in the coming months as well. Dan talked to the MJFF Foundation yesterday about those effects. So it's some promising markers that we're seeing in IPD, but we have a lot more work to do there.
It's work, great.
There's some questions in the chat. I don't know if we have more time, but there's questions in the chat. If you have time afterwards, maybe to answer them, please go ahead and do so.
We can take one more. We can take one more.
All right, well, Shalini asked, have you had success with measuring target and pathway engagement in the CNS?
Oh, in the CNS, yes. We will be talking about CNS target engagement in the coming months.
And there's one more question from Hugh Johnson at Toronto Western Hospital's Women's Disorder Clinic. Does therapy block aggregated alpha-synuclein, or is it preserving wild-type synuclein?
Oh, that's a great question. Okay, great question. So we do not affect soluble alpha-synuclein. And in fact, you don't really see a difference in soluble alpha-synuclein across the cell lines. What we affect is only insoluble alpha-synuclein.
Okay. Is there one other question?
Peter Lansbury has a question.
Oh, Peter Lansbury.
Peter's got his hand up.
Yeah,
I have a question sort of for everybody, because you really emphasize that 50% increase in activity is enough. So somebody who has a deletion on one allele, who has a very high risk of PD, will have GBA-PD, will only get to 75%. And so the question is, what is the data that supports your confidence that 50% is enough? And are you confident that, this is a linear thing? That is, that the more GCase activity, the more the symptoms will improve in a linear way. So even if you don't get it all the way normal, you'll get it most of the way normal. Those are related questions because and the third piece of that is, can you go to 450? Why not go to 100% activation?
Peter, it's a great question, right? There's a lot of that goes into that. One is that we're measuring Cmin activation, right? We want a minimum. So on the lowest concentration is 50% activation, which means during the day.
The Cmin Cmax difference is not all that great for this drug.
So we will be getting, when we model this out, we're at 75% activation. In fact, the dose we probably will take forward will be a minimum of 75% activation at Cmin, meaning you're at that AC 100 for a good portion of the time. This is also a new mechanism. We need to prove this. We have safety to do. So the dose selection is still ongoing. We have not tied ourselves to any specific dose yet. We're continuing to do, that's why we're running the 1B, is to look at target engagement, not just in healthy volunteers, but as you also, patients with GBA mutations, that is going out through 3 months. And we'll have those data and be able to choose a dose that really turns on GCase, but also with that's also consistent with a safety profile where we can take dose long-term.
Thank you.
Yeah, thank you.
All right. Stacey Henry from Denali is next.
Okay, can everyone see my screen?
Yeah.
Full screen? Apologies in advance, I'm getting over a cold, so my voice is a little dicey, but I'm really excited to talk to this group today about some of the work that we've been doing for developing a new therapeutic approach for GBA-PD. So my name is Stacey Henry, and I'm a senior director and staff scientist at Denali, where I oversee our lysosomal biology efforts. And so today I'm going to be telling you about our approach, which has been essentially developing a BBB penetrant enzyme replacement therapy for GBA linked to Parkinson's disease. So a little bit of background on Denali for those that are not familiar. Our main area of focus is developing therapies to treat neurodegenerative diseases. This includes neuronopathic lysosomal storage disorders, including MPS II or Hunter syndrome, as well as late onset diseases, including ALS, Parkinson's disease, and Alzheimer's disease. And as we think about what are some of the big challenges facing the field of neurodegeneration, one key barrier has been the blood-brain barrier as we think about how can we actually enable and enhance the delivery of drugs across the BBB to get to the key CNS cell types of interest that are affected across these diseases. And so our approach to try and overcome this challenge has been to develop a transport vehicle technology where we've essentially taken the Fc domain of IgG1 and engineered it to bind to the apical domain of the transferrin receptor. And by binding to the transferrin receptor, the idea is that it can essentially hitch a ride across the blood-brain barrier, as transferrin receptors normally are transcytosed across the BBB to deliver different ligands, including transferrin, into the brain parenchyma. And so by binding to the TFRs at the BBB, this can then bring different cargoes, including enzymes, across the BBB to the key CNS cell types of interest. So we have a pretty large enzyme portfolio using this technology. I'm going to talk just briefly about our most advanced program because I think this provides nice proof of concept that we think this can enhance the delivery of lysosomal enzymes across the BBB to the CNS, and then focus the majority of my talk on our efforts to pair this to glucocerebrosidase. So our most advanced program, as I mentioned, is for Hunter syndrome. We're currently in phase 2/3 studies and have submitted a rolling BLA. But we're really excited by this data. So essentially, in MPS II disease, you lack the IDS enzyme, which leads to the accumulation of its substrates, GAGs, including heparan sulfate. So these accumulate in the CNS. They can be measured in CSF, which is great because it provides a really nice target engagement biomarker to ensure that we are getting to the key tissue of interest. And in our phase 1/2 study in MPS II patients, we observed a normalization of substrate levels in the CSF of these subjects. And really excitingly, this correlated with biomarkers of disease showing a positive effect. We're able to normalize the levels of serum NFL in these subjects. So this suggests that this platform can enhance the delivery of lysosomal enzymes across the BBB and translate to meaningful benefit in terms of disease-linked biomarkers. So how are we applying this for GCase, and how do we sort of come to this approach? And so, as I mentioned, we have a generalist interest in lysosomal biology and a pretty broad Parkinson's disease portfolio. We're also working on LRRK2 and have LRRK2 kinase inhibitors that are in late-stage clinical studies. And as we think about, evaluating different targets to focus our efforts on, GBA has been a very attractive one, as has been mentioned earlier, based on the really compelling genetic evidence and the fact that we know that it is regulating key pathways that are linked to Parkinson's disease. And so I'm a cell biologist by training. As we think about what are the different therapeutic approaches that would be best tailored for the targets we're going after, it really comes back to the mechanisms at play. And for genetic targets, it's really, do we understand what the consequences of these genetic variants are to protein function, and how can we leverage that to guide our therapeutic strategy? So on the biology side, we first wanted to understand, how do PD-linked variants impact, in GBA1, impact GCase activity? So if you look at the different variant spread, there are some variants that are associated with Gaucher disease, including L4-4P and N370S. But a large majority of the GBA1 variants linked to Parkinson's disease are specifically associated with PD risk, but they're pretty poorly studied in terms of the impact on GCase function. So we focused on the E326K variant, which is the most common GBA1 linked variant in Parkinson's disease, and knock this into a cancer cell model and assess its effects on activity and protein function. And we could confirm that this does seem to be a loss of function similar to the Gaucher-linked variants. We saw a nice decrease in enzymatic activity that correlated with a significant increase in substrate accumulation as measured in a mass spec-based assay in these cells. So this suggests a common mechanism is occurring between both the PD and the GD-linked variants. How is it impacting GCase activity? Because that's useful as we think about whether we'd want to take a small molecule approach or a large molecule protein replacement-based approach. And so first we wanted to see, is this mutation just messing up the intrinsic activity of the enzyme itself? And so we purified recombinant wild-type GCase or GCase that has the E326K variant. When you look at the enzyme kinetics and parameters, it's really indistinguishable from wild-type. So we don't think that this mutation is killing the activity of the enzyme itself. But what about trafficking? And so for this, using our knock-in cell lines, we isolated lysosomes and looked at the levels of GCase in these cells. And as I think you can see in the E326K variants, which is pretty apparent, you see this really significant reduction in the levels of lysosomal GCase in these cells. So you're simply not getting enough of the enzyme to the lysosome where it can do its thing and clear a substrate. So as we think about going back to the mechanism, to us, this really suggests this leads to a loss of protein and would support an enzyme replacement approach for GBA-linked Parkinson's disease. So that's the strategy we've been taking. So to start, we fused GCase to our transport vehicle and wanted to confirm that we haven't broken the activity of the enzyme. It's still able to get into cells and clear substrates effectively. So we used a mass spec-based assay to look at the accumulation of the GCase substrate glucosyl sphingosine with increasing doses of our ETV GCase compared to the standard of care, which Lorraine had introduced earlier, imiglucerase for Gaucher disease. And so we saw a nice dose-dependent reduction with our enzyme. Fusing it to the transport vehicle doesn't seem to impair its ability to clear its endogenous substrate. Interestingly, we actually see that it's more potent than imiglucerase in these cells, and we think that is because by binding to the transferrin receptor on the cell surface, this enhances the amount that's being able to taken up and eventually delivered to lysosome. So that does lead to some benefit even in this cell model. Okay, so what about in vivo? So then we tested this in a GBA loss of function mice. Here we're using the GBA D409V mice, which have been made widely available by the Michael J. Fox Foundation. And we crossed this to a mouse model that has a chimeric version of the transferrin receptor that has the human apical domain and the rest of the receptor is mirroring. So it preserves the normal functions and binding partners for transferrin receptor, but allows it to bind to our molecule, which is designed against human transferrin. But we were pretty disappointed to see that this actually didn't lead to that much substrate reduction. So after a single dose, we were seeing about 27% reduction in glucosyl sphingosine in the brain. And as I mentioned, we have a pretty broad enzyme portfolio. Usually for other programs, after single dose, we'd see 50 or 60%. So this told us that there was something that needed to be further improved about GCase to enable this to be truly beneficial. And so then we try to figure out what's going on with this. GCase is known to be inherently unstable. And when we went back and actually looked at our molecule under different challenges, including, serum challenge with heat, we saw that the activity was rapidly killed. And this correlated when we actually looked at the amount of activity that remained following dosing in the mice. 24 hours post-dose, there's essentially no activity. So this is a bottleneck because we need to have enough enzyme that's circulating in the periphery to enable it to get to a high enough concentration to support receptor-mediated transcytosis and delivery to the brain. So to overcome this challenge, we took advantage of engineering approaches. So we used yeast surface display and a kind of clever assay to try and screen for variants that increase the stability and activity of GCase. So we started with greater than 1 × 10⁸ different GCase enzyme variants, used yeast surface display to display these variants on the surface of yeast, gave them a pressure, so trying to recapitulate what would happen in the body with, serum and heat exposure. And then using an activity-based probe in a FACS-based assay, we screened for variants that specifically led to an increase in stability on the surface and activity using this probe. And from this, we're able to identify a subset of variants that we then profiled in further studies. So first we wanted to see, does that actually increase and prevent this loss of stability in serum? So, for a wild type enzyme and the standard of care, as I mentioned, it's rapidly killed following heat exposure in serum. Some of our variants led to a modest boost, but we were able to identify some variants that led to essentially a protection of the activity following exposure to serum at higher temperatures. So this seemed to give us a way around this issue that we're having, and we were excited then to try and apply this to see if this could then lead to a more beneficial effect in terms of our ability to reduce substrate in the brain. And so going back to that mouse model that I introduced, we did the same study, but now in addition to the just wild type version of GCase, we tested our engineered version that we had identified through our yeast surface display experiments. And now we were able to see a really nice reduction in terms of glucosyl sphingosine in the brain. So when we dosed this mice, this is following a single dose, so just 7 days after a single dose treatment, we saw an 83% reduction of glucosyl sphingosine in the brain, which we're really excited about. And as we think about some of our other enzyme programs, we've never seen this much. We haven't engineered enzymes in the past, but I think this is really promising as we think about our ability to actually reach key cell types and clear the substrate that accumulate following GBA loss of function. We don't know the peripheral component of disease, but our idea is we also want to reduce substrate accumulation in the periphery. And as you can see, this also led to a really efficient reduction in terms of glucosylsphingosine in peripheral tissues, including the liver. And so as we think about potential applications for this, we are actively exploring both GBA-linked Parkinson's disease as well as Gaucher, because I think this also highlights their superiority to the normal enzyme and standard of care. And so this is something that we're actively exploring. So this is where right now we're in IND-enabling studies and really trying to focus on answering a few questions around, how does this correlate to correction of Parkinson's disease-linked deficits? Can we identify additional lysosomal pathway biomarkers in the mouse models to help understand how much recombinant GCase we really want in the brain to correct defects? Coming back to the question in the last talk that Peter had raised, we don't quite know how much we need to restore GCase levels to be beneficial. How much do we need normalization? Do we want to go past this? And so this is something we're actively exploring, as well as demonstrating that we're actually getting to the key cell types. For other enzyme programs, we've shown that you do get really nice distribution to neurons, microglia, and astrocytes. GCase trafficking, we also have LIMP2 in addition to mannose-6 phosphoreceptor and TFR. And so this is the big focus for us in our next stage. And with that, I'm happy to take any questions.
Any questions?
I'll ask one.
Okay.
Stacy, that was really nice.
Thank you.
But have you, so when I assume you're switching a little from glucere as a substrate to gluspig as a substrate. And, we looked a lot at that and the kinetics are very different. So now you're also switching the variant. So I wonder if you've looked at the variant that you're using, this new stable variant, to make sure that you're not just, well, it's a different question, right? Is the effect due to a change in the enzyme's turnover of gluspig, or is it just due to the fact that you're getting more GCase in the lysosome?
It's a fantastic question, and this is something we definitely want to explore, because one thing we were worried about was by engineering the variant, do we change the substrate specificity in some way that's not what we want? And so we've confirmed in cells, I don't think I had to add this into the appendix. I don't think I have this data in here. No, but we tested in cells to look at both glucosylceramide and glucosylsphingosine. We see significant, reduction of both. The potency is similar in terms. It's not similar. It's like 100-fold better still with the variants in these cells, but we don't lose any potency against glucer or gluspig. We've also used in vitro assays where we have glucer and gluspig in a mass spec-based readout to confirm that we're not losing that. And we don't see that we're now chewing up related glycosphingolipids. So at least from all of our cellular and in vitro characterization, it doesn't look like that we've impaired substrate specificity or lost any specificity for the substrates that we care about, or gained, degradation towards substrates that we don't want to hit. But it's a good question around the kinetics. We've been relying more on gluspig in our animal models because the glucer accumulation is pretty minimal, even in this severe D409V mice. We do have a colony of the E326K mice, to your point around the mutation, and so they don't have as much substrate accumulation, but we are interested in them because, at least based on our cell studies, that variant seems to lead to more profound lysosomal deficits that'll be interesting to look at. But it does seem like we're still hitting glucer, gluspig equivalently. We haven't explored the kinetics more. At least we seem, much more potent than wild type enzyme on both substrates.
Okay. Next up is Capsida. Thank you, Stacy.
Hi, everyone. Does everyone see full screen?
On full screen, yeah.
Wonderful. Thank you, Jonathan, and the rest of the foundation members for the invitation. I'm excited to go over Capsida Biotherapeutics' GBA-PD program with you guys today. I'm Nick Litsanis. I'm one of the co-founders and the Chief Research and Innovation Officer at Capsida. And Capsida is a next-generation AAV gene therapy company, so a little bit of a different modality than the majority of what we've heard so far today. You'll hear some similar themes, some different ones. But really, we are built around the premise that there is great promise to genetic medicines and gene therapy as a technology, but the first generation of gene therapy efforts have been met with a series of challenges that we believe sort of the cornerstone technology of Capsida, which is AAV engineering, is able to help surpass. And most of these challenges stem from an inability to get to and strongly engage targets, especially for neurological disease. And what that really looks like is wild type AAV serotypes like AAV9 being very ineffective at getting to the brain, transducing a large number of cells across the brain, and regardless, in many cases, of delivery technology. And what that results in is either very high doses needed for a variety of gene therapies that result in a number of safety complications, both in the liver, where a lot of virus accumulates, as well as in the dorsal root ganglia, in some efforts to sort of provide that virus through the CSF to get to the brain. And this has led to various toxicities in the gene therapy space, as well as more direct injection efforts into the brain having, of course, their own potential complications. Obviously, the invasiveness of the procedure, but also potential complications due to surgery, and an inability as well to use that route to broadly transduce large areas within the brain. So at Capsida, the cornerstone technology, as I mentioned, is the engineering of AAVs to pass biological barriers, to get to on-target areas more effectively and reduce the amount that gets to sort of off-target, non-therapeutically relevant areas. And what we've been able to do through a process of screening really large libraries in a high throughput manner of engineered AAV capsids is arrive at capsids that very effectively cross the blood-brain barrier, transduce the majority of neurons throughout the brain, and do so while detargeting the bioburden to non-therapeutically relevant areas in the case of many neurological disorders like the liver, like the DRGs. And doing this all with a convenient IV administration, single dose solution that provides a long-lasting ability, in this case, as we're going to talk about today, to provide a source of supplemental GCase enzyme to the brain. So around this technology, we built a fully integrated gene therapy company. We have not just these capsid engineering capabilities, we optimize cargoes, we have the capability to take this into the clinic, manufacture our own cGMP grade product, and do that across a variety of portfolio programs. The one we'll focus on today, of course, is CAP03, our PD-GBA program, where we are in the process now of wrapping up safety studies, our GLP toxicology studies. We expect to be moving into the clinic this quarter, first patient dosed next quarter, and some early biomarker readouts before the end of the year for this program. So the majority of the data I'll go over today is from that GLP tox program. But starting again, looking at why we chose Parkinson's disease, and specifically PD with GBA mutations, based on a lot of the great data that is in the field that others have spoken about today, we really believe that this is a great example of a space where the biology is beginning to get understood much better, but really sort of some of these first generation of efforts have just not been as efficacious at both testing this hypothesis in the clinic and providing the answer for really supplementing the activity of GCase that's necessary. And we've seen a number of different potential solutions for doing that today. Ours, like I mentioned, is by getting a source of GCase through a one-time therapy to the brain to be produced there directly in the cells. And we validated at this point that we can do that, achieve the levels of GCase enzyme elevation that would be expected to normalize, and much more than that, surpass the levels of GCase enzyme activity needed to supplement within the brain, and do that very safely without any safety events that have been seen throughout gene therapy so far. We've also validated at this point to the receptor that our novel capsids bind in the non-human primates. We know that those are conserved in humans and expect to see similar translatability there. Starting though with our cargo approach from rodent pharmacology studies in a disease model that sees drastic reduction in GCase activity and has overexpression of human alpha-synuclein. So we see increases in soluble and insoluble alpha-synuclein. What we've seen in the mice is, even though these mice exhibit a very severe phenotype, as you'll see here, GCase activity that's about 5% of normal levels in wild type mice, glucosal sphingosine that approaches 68,000% of wild type levels. We see that by providing, again, a source of GCase supplementation into the brain of these animals, you are able to drastically increase GCase activity, far surpassing wild type levels, reduce the glucosal sphingosine back to levels that are indistinguishable really from the wild type, and also see reductions in both soluble and insoluble alpha-synuclein at the same time. Moving to the non-human primate, though, testing our lead candidate that's now going to be moving into the clinic versus what has historically been used to try to get AAVs to the brain, which is delivery through the CSF. We see that the intravenous approach that crosses the blood-brain barrier very effectively gets to the brain, distributes and expresses within the brain. And what you see on this graph is both in areas like the frontal cortex, where some of these ICM, CSF-delivered approaches are able to get to with some degree of strength. We still see hundreds-fold increases in terms of expression through the IV route. But some of the deeper brain structures that are critical for the treatment of Parkinson's, that's now increased to sort of the thousands of folds. So much better expression throughout relevant brain areas for Parkinson's through this engineered BBB crossing route. And what that looks like from a protein expression perspective, here you see in purple expression of our therapeutic human GBA1 transgene, with an HA tag, which is what's stained here in green, various neuronal markers. You see widespread expression throughout brain areas. In substantia nigra, we're targeting about 60% of the dopaminergic neurons throughout other areas of the brain, ranging from 30 to 70%, and in other areas even higher. So what you really see is broad high levels of transduction, again, throughout the brain, specifically with this engineered candidate in neurons, which was the goal of the engineering process to begin with. And what that results in non-human primates, and now from here on, this is going to be data from our GLP tox study, is increases in total protein of GCase. I'll show you GCase enzyme activity and some of the target engagement biomarkers as well. These are all healthy wild type NHPs. So with that caveat, we're able to see very large increases in GCase protein throughout the brain. This is an average of various brain regions where across a dose response curve from 9.8012 VGs per kilogram administered IV up to 5E13 VGs per kilogram, we see from 50% up to over 400% of an increase in GCase protein over these wild type levels. And this is in comparison with, again, some of the earlier generation efforts providing a similar strategy through an ICM route of delivery with a non-engineered capsid, which are really capping out, we believe, at lower levels of expression than what's going to be necessary to treat the disease. What that translates to, importantly, in terms of GCase activity, again, over wild type levels here, is at the lowest dose here, 10% increase in activity, which we believe is subtherapeutic, up to a 200% increase in GCase activity. And I won't rehash some of the questions and answers given already, but we believe that this is going to be much higher than the necessary level to normalize and treat the downstream implications of the loss of GCase activity in patients. And importantly, as we now are moving into the clinic and looking at how we're going to be validating target engagement in the clinic, a very important pharmacodynamic biomarker for us is the activity and presence of our supplemented GCase in the CSF. So we've looked in the primates at CSF in our tox study. We see very large increases in CSF GCase protein levels. Again, up to almost 1,000% higher at the highest dose in this study than wild type levels. We also see that correlates very closely with the data I showed you on the previous slide, which was the activity levels in the brain tissue itself. So GCase protein in the CSF is going to be, for us, I think, an important biomarker of the pharmacodynamic effect of what's happening in the tissue of the brain specifically. We've also looked at glucosylsphingosine in these primates. Again, these are healthy primates, so there isn't an elevation of glucif. We're actually unable to detect it in the CSF at all. So here is sort of some of the data from plasma that shows that there's modest decreases in glucosylsphingosine, again, even in this wild type background. We saw from some of our pharmacology studies that that's the case in mice and the mouse brain as well in a wild type context, but of course in a diseased context, when those are greatly elevated, we see very drastic reductions in glucosylsphingosine. So we expect a similar effect in a disease context in primates and humans as well. And then very importantly, from a safety perspective, what we saw in our GLP toxicology study, as I mentioned up front, is very drastic reductions in some of the off-target distribution. Compared this to historically to AAV9, which delivers to a high degree to the liver and other off-target areas in the body, even at a higher dose here, this is our 5e13 dose, so double the dose of what we've tested of AAV9 in the past. We see almost a 20-fold reduction in distribution to the liver. We see a reduction in expression in the DRGs. And what that all really leads to at the end of the day is a very well-tolerated safety profile that we saw in our tox study. We saw no adverse events throughout the 3-month tox study and at any of the doses that we saw. And so we expect not only a great efficacy profile, but a great safety profile as well now that we move into the clinic soon. As I mentioned also up front, we have in-house manufacturing, which is critical in the gene therapy space. So not just process and analytical development, but the ability to scale up our product. We've now made product for the upcoming clinical trial and expect to be able to continue to support that trial through into later phases. And just finally, to talk about where we're at today, as I mentioned, we expect to be dosing first patient next quarter. Expect to start to get some of those early biomarker readouts before the end of the year. We look to be entering a patient population with confirmed GBA genetic diagnosis with Parkinson's disease, and will, of course, be measuring a variety of similar outcomes and scale measures as some of these other programs. I'll pause there and see if there's any questions.
I had a question for you. Thank you for the presentation. Pretty exciting data, and just a question on what do you think is driving the differences in transduction percentages in different brain regions?
I think to some degree it's density of cells, Dan. The cortex is one of the most dense regions. We believe there isn't really a difference in BBB crossing across different brain areas, but a similar sort of distribution of virus is going to result in a smaller number of cells transduced if it's a more dense population. So we think that's one of the main drivers.
So you don't think it's kind of a neuronal subtype or something that is more likely to be transduced than others?
We haven't seen, I think, anything so far that suggests that it's a cellular subtype difference. We know we transduce neurons specifically, but within that population, not really.
All right, thanks.
Any other questions? All right.
One question, maybe.
Sure.
Oh, sorry.
No, go.
Just a quick one regarding also the patients that you're including. Do you believe or do you suspect that specific GBA mutation may have an effect on the outcome? Yeah, is it something you look at as well?
We are not planning to enroll only specific mutations as a first step. I think the verdict is still out there, right, on whether or not specific mutations will react differently. So I don't want to hypothesize at this point if that'll happen, but we're not expecting to go into a subset of genetic mutations at this time.
Right, thank you.
All right, thank you very much. Is there another question? All right, let's move on to Gain Therapeutics. Jonas, are you around?
I'm here, yes.
Great. Can you share your screen?
I will. Let's see. Is that displaying for everyone?
Yep.
Yep. All right. So I'm Jonas Hannesad. I'm the Chief Medical Officer at Gain Therapeutics. And I'm going to talk about GT-2287, which is our small molecule treatment and development for Parkinson's disease. Here are forward-looking statements. We're a publicly traded company. And just brief background on the company. So Gain Therapeutics was a spin-out from a Spanish company called Minorix in 2017 and was founded in Switzerland. And then we had an IPO in 2021 and we're listed on NASDAQ. We have right now about 26 full-time employees and they're distributed between the US, where headquarters is, and Switzerland and Spain. And the technology that enabled us to develop this lead compound is it's a computational platform called Magellan, which models the interactions of proteins, and in this case, enzymes and small molecules. And then that's used as a starting point for then synthesizing these molecules in real life and characterizing them. And that's how GT-2287 was discovered and developed by using this computational platform to model glucocerebrosidase interactions with small molecules. So we have completed a phase 1 healthy volunteer study completed last year, and we are currently conducting a phase 1b study in people with Parkinson's disease, both idiopathic and GBA1 carriers. So just brief background on some of the in vitro and vivo effects that we've seen with 2287 before I move on to the clinical parts. So I think everybody on this call probably knows this, but the GBA mutations is the most common genetic risk factor for Parkinson's. It increases your risk between 3- and 5-fold, depending on the mutation, the population, et cetera. And it's also associated with sort of I would say, more aggressive phenotype, right? So it's an earlier onset, faster progression on average, and more cognitive decline, unfortunately. Now, several of you mentioned earlier the hypothesis as to why GBA1 mutations increase your risk of Parkinson's, and there's a loss of function, gain of function, various hypotheses. We think that while the loss of function of enzymatic activity in lysosomes, as Kevin was speaking about, that is a very important component, but we don't think that's the only component. We think there's also a gain of function, I guess you can call it, and then the other pathways in the cell are involved. And that is the reason we chose, instead of developing an activator specifically, we've developed a chaperone, a stabilizer of GK, so that you increase the amount of protein and you reduce the amount of misfolded protein. So what we know about GCase and GBA1 in Parkinson's is that it clearly reduces GCase activity in lysosomes, right? But in addition to that, you have a lot of endoplasmic reticulum stress from the fact that you activate the unfolded protein response when you have misfolded protein, GCase in the ER. And you also have impaired mitochondrial function. And possible explanation for that is that, and this was recently discovered, GCase has a role also in mitochondria. It stabilizes complex 1 of the electron transport chain. And that appears to be a structural function, not an enzymatic function, so different from what happens in lysosomes. And we've shown that with our approach, stabilizing the protein, we get more enzymatic function, better lysosomal function, but also better mitochondrial function. And those approaching both of those pathways then leads to increased neuronal survival and less neurodegeneration, both in vitro and in vivo. So what we've seen is, so we've used patient-derived cells, both carrying mutations and not, and the mouse models where we model both GBA Parkinson's using inhibition of GCase and just idiopathic Parkinson's just using alpha-synuclein preformed fibrils. In both in vitro and in vivo, we can demonstrate all of these effects. So reduce alpha-synuclein aggregation, reduce ER stress, improve lysosomal function, reduction in the substrates of GCase, and improve mitochondrial function, and also reduce neuroinflammation, because that seems to be a sort of a spillover effect from these, what happens inside the cell. And all of these effects together then appear to come together somehow and improve the function of these neurons and decrease their degeneration, and improve in the mouse models, improve various behavioral phenotypes that are result from these insults. So just quickly, because I know we're short on time, but here are just some examples of what we see, in vivo, that we have increased GCase activity, reduced ER stress, improved lysosomal integrity, better mitochondrial health, and then reduced aggregated alpha-synuclein. And one aspect of GBA1 Parkinson's, and idiopathic Parkinson's for that matter, is that's obviously very important to patients, is cognitive impairment. So that's probably you could argue that it's more debilitating than the motor symptoms. And what we've seen is we've tested this sort of as a, on the side, whether our approach has an effect on cognitive function as well. And what we used to do this is a model where we look at nest building. So mice have an instinctual behavior, they build nests. And you can impair the nest building either by giving CBE, which is a pharmacological inhibitor of GCase, or by giving preformed alpha-synuclein fibrils injected in the striatum bilaterally. Both of those insults will impair the mice's ability to make their nests. And then when we treat with a compound, whether it's the CB model alone, CB plus PFF, you see here in the dose-dependent manner, you sort of recover their ability to build their nests. So that, that could be, I guess, seen as a pro-cognitive, or, helping the mice improve their planning ability and their sort of spatial cognition. So moving on to our clinical development program, as you know, you ask different questions at different stages of development, right? So in the phase 1 study in healthy volunteers, typically what you want to know is whether your compound is safe and well tolerated, whether it has adequate pharmacokinetics, and whether you can get target engagement, right? And that's what we demonstrated in our phase 1 study. So this was a pretty standard single ascending dose, multiple ascending dose study in healthy men and women ages 18 to 65. We decided to include about 15% of individuals older than 50, because ultimately most people with Parkinson's are middle-aged and older. And we had 5 cohorts in the single dose part, and the dose levels are depicted here, and then 4 in the multiple dose part. And these were dosed daily for 14 days. And in addition to safety and tolerability and plasma pharmacokinetics, we also looked at CSF levels of drug just to confirm that it gets into CSF, which we had looked at that in rodents, and also GCase activity in blood and whole blood. And we presented these data elsewhere, so I'm not going to go into details, but we show that 2287 was safe and well tolerated, both a single dose and multiple dose for up to 14 days in healthy volunteers. We had no serious adverse events, no grade 3 or severe adverse events. And I think it was about 90% of the adverse events were mild, so very few moderate adverse events. And we had no discontinuation due to adverse events. And with regards to safety parameters such as ECG, labs, vital signs, et cetera, we didn't see any clinically significant changes there that would indicate a safety signal. And this is fully consistent with what we've seen in toxicology studies, so in rats and dogs. There's really no safety signals in any of the histopathology whatsoever. We have plasma exposures in what we project is the therapeutic range. So this again is based on in vitro and in vivo data. We can get to those levels readily with the oral administration. And we have adequate safety margins with regards to tox studies. We did demonstrate that 2287 gets into the CSF, which presumably gets into the brain. And we know in rodents that levels in brain are about several fold higher than in blood or in plasma because of the physical chemical properties of this compound. And lastly, we did demonstrate an increase in GCase activity in healthy subjects in dry blood spots. So after completing this phase 1 study, we then started a phase 1b study in patients. And the key questions we are asking in this study is it safe and well tolerated in people with Parkinson's who are generally older than the healthy volunteers we enrolled, and for longer duration of dosing, so for 3 months instead of 2 weeks. In addition to that, we're obviously very interested in whether the compound can modulate the relevant biology in people the similar way to what we see in mouse models, for instance. So in this study, it's an open label study. So all the subjects are getting active drug. They get 13.5 milligrams per kilogram per day for 90 days. And we're aiming to enroll between 15 and 20 people. And they have visits, baseline being 14, 30, 60, 90, as you can see here. The population we're enrolling is, they have a clinical diagnosis of Parkinson's disease, diagnosed within the last 7 years, so early to mid-stage Parkinson's, and they can either be idiopathic or GBA1 carriers. And we are attempting to get about half of each so that we can compare the biomarker responses in those two populations. And people can be on stable dopaminergic treatment or treatment naive. As I said before, the primary endpoint is safety and tolerability and pharmacokinetics, but then the biomarkers that we're evaluating include GCase activity, as we did in the healthy volunteer study, levels of substrate, so glucosylsphingosine and ceramide in both blood and CSF, inflammatory cytokines, because we see very clear anti-inflammatory effects in our mouse studies. So we're looking at that both in blood and CSF as well. We are measuring alpha-synuclein. We don't know if alpha-synuclein will change in a 3-month study, but we're measuring that. And then we're measuring various markers of lysosomal function, mitochondrial function, because we think the mitochondrial part is also an important aspect of this mechanism. Skip this in the interest of time. This is the study design. So as I said, screening period, and you have 13.5 milligram per kilogram. In case somebody has tolerability issues at that dose level, they're allowed to decrease to 11. Nobody has done that so far, but that's just to make sure we keep people in the study should some of them experience, adverse events or tolerability challenges. And so in summary, Gain Therapeutics is developing a stabilizer, a novel small molecule that stabilizes GCase protein and enables more of it to go to lysosomes, where GCase enzymatic activity is important, but also to mitochondria to improve mitochondrial dysfunction. And we have shown that this compound is safe and well tolerated in healthy volunteers at doses that produce therapeutic plasma levels both in blood and CSF. And we're currently evaluating this compound in people with Parkinson's, both GBA1 carriers and idiopathic. And we should have preliminary data from this study sometime this fall. And then depending on those data, we're planning to initiate a phase 2 study in Parkinson's sometime early next year. So thank you.
I could ask a question, Jonas, please. Could you share the brain to plasma ratio across preclinical species and what you're seeing in CSF versus unbound plasma?
Yeah, so in humans, it was like 1.03. I think the ratio was like, you know, just about 1. So this compound is it's highly, you know, highly penetrable with a low solubility. So it has very low levels in CSF because it doesn't like water, basically. It's
yeah.
So in mice and rats, levels are low in CSF, levels in brain parenchyma are very high, but we mirror that in humans. So levels in CSF that we saw in the healthy volunteers is basically in line with what we saw in mice and rats. And you know,
so the CSF to unbound plasma is at unity.
Yes.
So in human. Okay, excellent. Good. Thank you.
Any other questions?